All right, in chapter 19, we are going to start our discussion of various diseases, and I'm mainly using these diseases to highlight things we've already learned about. So to highlight some of the different toxins or unique structures to a microbe or to a virus that allows it to cause infection, as well as just using some of our other terms as far as normal flora and our immune defense. So again, they're meant to provide information about various diseases you're likely to encounter. We're not covering the entire chapter and all of its content, to really focusing on using your study guide and the PowerPoint to help kind of hone in on the important information. So first of all, starting with our natural defenses. So this chapter covers skin and eye. So starting with the skin first, we have what's called the integument, and that includes the skin, our hair and nails, as well as sweat and oil glands that are able to produce things that naturally prevent an infection. And they just provide that barrier, that first line of defense that we talked about in chapter 15 that prevents things from getting in. In addition, there are antimicrobial peptides. They get made by the skin, and these are positively charged chemicals, and the membranes, the phospholipid bilayers, are negatively charged. So those positive charge kind of disrupt and dismember the membranes of a lot of microbes. In addition, a few other things. Sebum, which is made by the sebaceous glands. This has a low pH, so it keeps the skin nice and acidic, makes it inhospitable for a lot of microbes because they like a neutral pH. But also, of course, there are organisms in the normal microbiota of the skin. The sebum also provides a lot of fats and a lot of lipids that provide nutrients for them. Then sweat as well has that low pH that helps produce that acidic nature, but also a high salt concentration. Again, that inhibits a lot of microbes because they don't like the extra salt. And then finally, lysozyme that we mentioned a few times. This is a chemical found in sweat, tears, and saliva. So it's part of our natural defenses because it breaks down peptidoglycan. And if you remember, this is going to be more harmful to gram-positive bacteria because they have a thicker peptidoglycan cell wall compared to gram-negative. So especially with the eye, whenever we're talking about the tears, a lot of bacteria that cause eye infections are gram-negative because the lysozyme keeps a lot of gram-positives away. Now, the normal biota on the skin, again, overall, the skin is inhospitable due to, again, it's dry, also it's salty, those antimicrobial compounds, it's acidic as well. So again, where we find them, so we're going to have a denser population of microbes where it's moist, so the folding of the skin, also the hair follicles and the various ducts that release those oils in sweat. The two most common microbes that are part of the normal flora of the skin is Staphylococcus epidermis. So staph like salt, epidermis referring to our skin layer. And then the one that causes lots of acne problems, the Propionibacterium acnes. So these are the two most because they can tolerate that high salt. And the majority of them live in the top part of the epidermis, so they're not covering down into like the dermis of the skin, and about a quarter of them are found in the hair follicles. And again, that's what leads to like infected hairs and that type of thing. All right, so our couple that we're going to focus on here. So first starting with Staph aureus. So Staphylococcus tells us that grape-like structure. Again, that's some of our terminology about the arrangement. So again, coccus means that they're circular, and staph indicates this grape-like clustering effect that we get. So just from the name kind of gives us an idea about that. It is gram-positive, so it is sensitive to that lysozyme that destroys that thick peptidoglycan layer. Doesn't have a flagella, so it's non-motile. They do not form endospores, but of the bacteria or of the pathogens that don't form endospores, which are most resistant, this is one of the sturdiest microbes. So whenever we're talking about MRSA and VRSA that are extremely antibiotic resistant, this is why it's a problem. These are very hardy organisms. They're hard to kill because they can withstand salt concentrations, changes in pH, and even high temperatures. So it makes them very hard to kill. Again, they're not at the endospore level, but they are very hardy. These can also be viable for months, not moss as it says there, but for months while they're outside. All right, now again, MRSA is the main reason that we talk about this one, as well as some other soft tissue infections, but these cause these skin lesions that we see over here. Again, they're resistant to multiple antibiotics, which is why they're a concern. They cause these kind of raised lesions here. There's usually red around it. It's localized to one location, and a lot of times there's pus inside of here as well. Typically get fever along with these, and these are going to occur when there are breaks in the skin, because again, they need to be able to get through that first line defense. So they rely on breaks due to injury or shaving, or just rough skin, or abrading against that tissue. Now, what makes Staph aureus so pathogenic? So think about virulence factors and things that help it become pathogenic. It produces lots of enzymes that we'll talk about here. So coagulase is one that is involved in coagulating plasma. This is the test that you see over here. So it's actually able to break down the red blood cells. So whenever you put it on blood agar, it creates a clearing here. Hyaluronidase, this is common. It breaks down hyaluronic acid that helps connective tissues bind together. So by breaking that connective tissue, it just produces separation and allows for further infection. So it's removing some of that natural defense by breaking down connective tissue. Staphylokinase gets rid of blood clots. So again, breaking down that plasma, breaking down clots. DNase can be produced that destroys DNA, harming our own cells or other microbes to help it compete. Lipase, it helps break down or use lipids, so that's why it can exist on oily skin. It's actually breaking down those lipids that normally are harmful. And it produces catalase, which is able to break down peroxide, so it converts it into water and oxygen. And this is actually the main test that's used to distinguish Staph aureus from Streptococcus pyogenes that we'll talk about in a little bit. So this catalase is made by Staph aureus, but it is not made by strep. So a lot of the diagnosis goes along with determining which virulence factors these various organisms have. All right, so there are a few summary tables throughout. You don't have to know all the information on here. Of course, the causative agent, a little bit about the transmission. So is it direct, so touching, or indirect, so picking it up like off of a doorknob or something. Talking about the virulence factors, so we just named all those different enzymes that they can produce. I'm not concerned that you know about the culturing so much. Again, the catalase test, that helps identify organisms that can produce that enzyme versus others that can't. Again, like the Streptococcus pyogenes we'll talk about. As far as prevention, again, thinking about MRSA, and this is one of the main ones that we worry about in hospitals. So good hygiene practices are instrumental with preventing infections, and especially from the spread of infections. So again, hand washing, sterilization, all of that is so key to preventing this, not only in healthy, but also in healthcare facilities. As far as treatments, again, it's limited to the antibiotics, and this is why it's a serious threat based on the CDC. So it is something of concern in the healthcare system. Now switching into impetigo, which is another skin infection. Again, this is superficial, so just on the outside of the skin, it causes this kind of flaking and peeling off. Now this can be either caused by Staph aureus we just talked about, or Streptococcus pyogenes that we'll talk more about here in a minute, or even the mixture of the two. And this is usually found in kids. And the symptoms look the same whether it's caused by Staph aureus or Strep pyogenes. It still is going to produce this kind of scaly, dried out, and kind of skin infection that we see here. So again, that caused by Staph aureus. We already mentioned some of the enzymes. It also produces exotoxin, which creates an exfoliative toxin. So think about exfoliating our skin, which is like sloughing off that rough outer layer. We do that with a lot of health, like a lot of skincare. But this toxin is how it's able to actually attack it. So it attacks the epithelial cell-to-cell connections, and it causes this blistering effect. So it actually separates the dermis and the epidermis, and it causes that fluid-filled blistering to occur due to that toxin that Staph aureus can produce. And we already mentioned, though, the coagulase that coagulates the plasma in the blood. So this causes fibrin to get deposited, and it concentrates that exotoxin into one place so that it causes local damage, again, leading to that blistering effect. Now, the Streptococcus pyogenes, the other one that can cause the impetigo, also gram-positive. Again, coccus tells us this, but instead of being a cluster, it's going to be that chain. We can tell by the naming there. It is beta hemolytic, meaning it can break down red blood cells. Now, in addition to causing impetigo, it can cause multiple other diseases as well. So strep throat is a common one. If left untreated, that can also lead to scarlet fever. Strep pyogenes can cause pneumonia, so we'll come back to that when we get to respiratory diseases. Purpura fever, necrotizing fasciitis, so like flesh-eating disease, various bloodstream infections, and rheumatic fever. So again, sometimes the organisms we're talking about can cause multiple infections depending on where they're at. Now, some of its virulence factors is its ability to adhere. So it actually coats itself with host protein, so it doesn't look foreign, so it evades the immune system with those M proteins. It also makes that hyaluronic acid. Again, it can break down the connective tissue, the connection between connective tissues, so just allows it to spread and cause disease. So then how do we identify some of these? Because these are so often used, we won't talk about them for all organisms, but especially those common in healthcare. So the rapid strep A test, so this is usually looking for like strep throat, but it can identify other conditions as well. But again, there's a rapid test that produces a result very quickly. But then also, if it needs to be confirmed, or if it, they're not sure about the results of the rapid test, they can send it off for culture. And what they're looking for is that beta hemolysis. So they're going to grow the culture on a blood agar plate. So it just has a bunch of red blood cells. And if it is strep, it produces this beta hemolysis. And it's where the bacteria grows. You can see the little dot in the middle, but it actually breaks down or eats away at that blood. So, and then a certain pattern, it leads to this beta hemolysis detection. And then at the same time, it's also sensitive to bacitracin, unlike some other common causes of like sore throat and stuff. So another confirmative test is putting a disc that has bacitracin antibiotic on it. And if bacitracin is able to kill it, then it indicates that it is strep. Now, bacitracin doesn't kill staph, so that's a way of telling the difference between strep and staph besides the catalase tests. All right, so just kind of our summary table here for impetigo. Again, it can be caused by either Staph aureus or Strep pyogenes or both. Again, both are from direct or indirect contact, so skin-to-skin touching or picking it up from like a doorknob or something. As far as virulence factors, again, that exfoliative toxin, coagulase, and all the enzymes we've already mentioned, like the DNase and catalase that were mentioned. Now, Strep pyogenes produces streptokinase, plasminogen binding ability, just meaning it can bind to that. The hyaluronidase, the M protein is what it uses to kind of coat itself to look like it's a normal self cell instead of a foreign pathogen. Again, not worried so much about this culturing and diagnosis. Again, hygiene practices is what's used for prevention, but both of these have alerts from the CDC. So again, Staph aureus is a serious threat because of that MRSA. And then the strep pyogenes is also a concerning threat, so not quite to the same level. But again, it's starting to develop that antibiotic resistance as well. Switching now to gas gangrene, this is one that I like to talk about because it's a spore former. So gas gangrene also important in healthcare because that's why it's so important that wounds are treated effectively. This is also called Clostridial myonecrosis because it's caused by Clostridium perfringens. This is again one of those spore formers. It's also gram-positive, but it has the ability to produce those spores that make it really hard to treat, and why, again, proper cleaning is important because those endospores are found in soil. So again, if you have some type of injury and it doesn't get irrigated and cleaned out well, those endospores hang out in there, and then once they're inside the body, they start to germinate and produce an infection. They're also found on skin. They can be found residing in the intestine and the vagina as well. So again, these endospores just really, really hard to treat. Now they do require anaerobic conditions. So as we'll see, treating them, again, just leaving them open to air so that it can be aerated because they have to survive where there's no oxygen. So again, providing oxygen and keeping wounds open when possible, as well as making sure they're irrigated and flushed properly, is extremely important to prevention of gas gangrene. Now a little bit about this, or a little bit more about this organism. Again, it's gram-positive, it's a spore former. As far as virulence factors, it also produces exotoxins. Again, these are toxins released from the cell. And those include an alpha toxin. This causes red blood cells to actually rupture. So we have beta hemolysis and alpha hemolysis. This is one that produces that alpha. It can also cause edema, so that's swelling and tissue destruction. The collagenase breaks down collagen in our skin. Again, it allows it to penetrate through that layer. The hyaluronidase that breaks down connective tissue, and the DNase to break down DNA of other cells. Again, our table here, again, gas gangrene can be caused by a few others, but Clostridium perfringens is the most common, so that's what we're focusing on. Again, as far as modes of transmission, it's somehow being able to get into the skin, so using soil, going through an opening or an injury. Again, those virulence factors like the alpha toxin and other exotoxin. It forms that gas because, again, it's anaerobic, so as it's going through respiration, getting food and energy, it releases gas as a byproduct. Again, not so much concerned about the culture. Again, prevention, getting rid of dead tissue, which is called debriding. So removing that dead tissue, making sure wounds are clean. And then as far as treatment, again, oxygen can be used because it has to be anaerobic, so keeping it open. Again, as well as getting rid of that dead tissue and surgical removal and some antibiotics. Now this table and on the next slide is showing some of the terms that we used when talking about these skin conditions. So a bulla, these are like blisters, so these are going to be large, wide vesicles. A cyst, so this is raised and encapsulated, but solid or semi-solid. So it's not going to have like a bunch of pus in it, but it's going to be in a harder state. A macule, this is a flat, demarcated lesion, usually indicated by some type of color change to it. So like a freckle, for example, could be an example of a macule. And then kind of putting a couple words here together, maculopapular rash. So this is flat, maybe a little raised bump to it. So like measles and rubella have these rashes we'll talk more about. A papule, So these are small, elevated, again, small, solid bumps, like a wart would be an example there of a papule petechiae. These are small purpura that's on the next slide, but kind of like a small, teeny tiny rash, little red sites that we get on our skin. A plaque here, elevated flat top lesion, but it's just larger than the papule is the only difference. So it's still kind of solid, but like plaque cirrhosis, you'll hear about advertising on TV for different ads. So again, just a larger area, but it's a solid, slightly elevated lesion. The purpura that we mentioned, so this is kind of reddish purple discoloration due to the blood in the area. But it doesn't blanch whenever you push on it, just meaning it doesn't turn red. And then whenever you push on it, it'll go to white. So it stays red the whole time. A pustule, as the name kind of implies, it has pus inside it. So it's an elevated lesion, but instead of like a blister that has a clear fluid, this has pus inside of it, indicating some type of infection. A scale, talking about the flaky portions of the skin here. And then a vesicle, just an elevated lesion, kind of like the pustule, but it has a clear fluid instead of the pus fluid.